like before ancient DNA, we were using modern DNA to make inferences about populations, their history, their demographic history, and we were making inferences about genome evolution using modern DNA. But with the discovery of ancient DNA, or with the use of ancient DNA, the whole area has changed, because we could directly use the archaeological remains from the past populations and then make direct inferences about them, about their population history, about their demography, like if the populations expanded or shrink, or if there has been some human, or it doesn't have to be human, could be any organism. If there has been any movement around, which means if there has been gene flow between populations. So we could make these direct inferences using ancient DNA. That's why ancient DNA has been a revolutionary change in our studies. So first, I would like to talk about a very striking example where ancient DNA made a huge difference in the area of like archaic human population histories. So we're all familiar with our cousins, Denisovans and Neanderthals and modern humans. We know that like only about 50,000 years ago, we were sharing the earth with our cousins. We were all living at the same time and our ranges were overlapping. We know this from the archaeological evidence, right? From the archaeological remains. But what we didn't know was if they did interacted, if there has been matings, interbreeding between these archaic humans and ancient modern humans. So that's where ancient DNA comes into play. So we could use ancient DNA and get the genomes of Denisovans and Neanderthals and ancient modern humans and compare them and see if there has been some gene flow, some gene transfer between the genomes of these human species. And ancient DNA studies showed that yes, there has been matings or there has been interbreeding between all of these human species, between modern humans and Neanderthals, modern humans and Denisovans, as well as Denisovans and Neanderthals. So this figure here shows the gene flow between modern humans and Denisovans, modern humans and Neanderthals, as well as Denisovans and Neanderthals. And these numbers are the amount of gene flow or amount of the genome shared between these groups. For example, like there has been gene flow between Neanderthals and modern humans, and like about 1.5 and 2.1% of our genomes basically inherited from Neanderthals. But that's between Neanderthals and, I should mention, non-African populations, Eurasian populations specifically. So, I mean, these numbers tell us that, yes, there has been gene flow between all of these human species. So this was really striking. I mean, as I said, from the archaeological evidence, we knew that they shared the habitats, but we didn't know if they mated. Another striking result was the maternally inherited mitochondrial DNA. This is a molecule, a circular DNA molecule found in all of our cells, and we inherit it from our moms, which means moms pass it to their children, but dads do not. Okay, so we can trace like the maternal lineage by looking at the mitochondrial DNA. So as a result of these studies on Denisovans, Neanderthals, and modern human, they also saw that maternally inherited mitochondrial DNA and Y chromosome, which is specific to males, mitochondrial DNA and Y chromosome coming from the modern human replaced the Neanderthal mitochondrial DNA and Y chromosome as a result of these matings. So these were really like striking and surprising results in the ancient DNA research area. So again, another study involving Neanderthals and Denisovans showed mating between Neanderthals and Denisovans. So in this map, the blue individuals are the Neanderthals, and then the yellow ones are modern humans, and then the red ones are the Denisovans. So there is this one individual called Denisova 11, who they found to be a 13-year-old girl, was apparently the offspring of a mating between a Neanderthal mother and Denisovan father. So they found this bone, finger bone, and they extracted ancient DNA from this, and they obtained the genome and compared it with Neanderthals and Denisovans, and they found that it carries signatures both from Neanderthals and Denisovans, and its mitochondrial DNA, which is inherited from mother, was belonging to a Neanderthal lineage. So they could tell this was a mating between Neanderthal mother and Denisovan father. So, I mean, by using ancient DNA, so we can make like inferences about the matings, population history, or population dynamics. But it's not only the population-related events. We could also make some inferences about like what kind of genes were inherited from Neanderthals and what do they code for. So, you know, we got some Neanderthal genes into our modern genomes. So, but what were these genes? What they were coding for? So research showed that some of those genes were linked to modern diseases we have in the present day. For example, like neurological diseases, depression. Apparently, depression is related to the Neanderthal genes we inherited from them. Also, addiction to tobacco, smoking habit was linked to the Neanderthal genes. Some other diseases related to like cardiac diseases, digestive diseases, or some urinary tract disorders, they're also linked to the Neanderthal genes. Again, like predisposition to skin cancer. It is also related to the genes we inherited from the Neanderthals. I mean, it all sounds like bad and negative, but we haven't inherited all of these bad traits from Neanderthals. We also have some good genes, like genes boosting our immune system. Apparently, they're also inherited from Neanderthals. Also, genes increasing the fertility were found to be linked to the Neanderthal genes. So this, I mean, all of these fascinating results were obtained as a result of ancient DNA studies. Of course, this is not our area of study in ODTÜ and Hacettepe labs. We're working on modern humans, not these archaic humans. But I just want to show you what ancient DNA can do, can tell us about populations, their history, and also like the genome evolution. Okay, so I think after this striking example, I should talk about what ancient DNA is, how we obtain ancient DNA, what are some sources of ancient DNA. Ancient DNA is the DNA obtained from archaeological remains or museum specimens, as you can see here. Like remains older than 100 years old. DNA obtained from those remains are called ancient DNA. Anything younger than 100 years old is called historical DNA. Okay, so we can use museum specimens, bones from excavations, and those could be like cranial bones or postcranial bones or tooth Sometimes coprolites, which are fossilized feces, they can be a good source of DNA, both DNA source belonging to that human and also the microorganisms colonizing the body of the individual. Hair, which is kind of rare and difficult to come across, but also is if left, if can be accessible, then is a good source of ancient DNA. Aside from these, there is some indirect sources of ancient DNA. For example, sediment DNA. DNA trapped in the sediments in caves or in archaeological excavations. So people collect sediment samples and then extract DNA out of them. Of course, there is not going to be only organisms which lived in that habitat, but there will be microorganisms. Whatever got trapped in that sediment will show up in your DNA soup. So, but these are the major sources of ancient DNA. But why we call it ancient DNA? How is it different than modern DNA? How do we diagnose if it's ancient or not? Of course, there are certain characteristics. First of all, DNA gets fragmented during time, fragmented into small pieces when it's exposed to heat, acidity, and humidity. If it stays under the soil for thousands of years, of course, it will be exposed to temperature fluctuations. Sometimes, rainwater will seep into the soil and then will wet the bones. But soil itself is humid anyways. So being exposed to that humidity and then acidity fragments the DNA into pieces in time. The longer it stays outside or in the soil, the more fragmented it gets, okay? Another characteristic of ancient DNA we obtain is it's contaminated. It's contaminated with modern DNA. For example, from the excavation personnel, the anthropologist or archaeologist excavating the bones can contaminate, or the researchers in the laboratory who are studying on these samples can contaminate ancient DNA. And also the microorganisms, bacteria that colonizes the bones after post-mortem can contaminate the ancient DNA. So, different from modern DNA, it has large amounts of contamination. And another very diagnostic characteristic of ancient DNA is, again, as it sits in the soil or in the excavation house, if it stays there for a long time, then chemical changes start to happen. The cytosine bases, which is found in the DNA composition, changes into another base called uracil. It changes into actually thymine. So we see these cytosine to thymine changes, which is diagnostic for ancient DNA. So in summary, it's biochemical characteristic changes. So that's how we diagnose the ancient DNA in our studies, okay? When we go back to the beginning of the ancient DNA studies, now I would like to give you some chronological perspective from the beginning of the ancient DNA studies to today. What were the first ancient DNA studies done, and then what happened till today? So the very first ancient DNA studies were done on this extinct member of equid family. This species is called quagga. It's extinct now, and the study was done on a museum specimen. And at that time, we didn't have technological tools to, sequence or access to whole genome. Like in 1980s, which was the beginning of the ancient DNA studies, people were mostly looking at those mitochondrial DNA molecules and sequencing small parts of the mitochondrial DNA, because that's what the technology would allow us to do at that time. So the another ancient DNA, the earliest ancient DNA was done on an Egyptian mummy. So again, it was done on a small piece of mitochondrial DNA. But throughout the time, the number of ancient DNA studies increased. By the way, this is mitochondrial DNA. As I mentioned before, it's a circular DNA molecule, and we can sequence small parts of it and reach this genetic code and compare it with other individuals and see like what kind of differences there are between individuals. And we can make some inferences about the populations using these sequences. So, initially we were using, or people were using mitochondrial DNA in their studies. Okay, so after the 1980s, during the 1990s, we saw a great increase in the number of ancient DNA studies. And the peak reached when a group said they sequenced DNA from an 80 million year old dinosaur. So, this was like the top point of ancient DNA studies. But it was just, they sequenced the DNA, published it, but they haven't done any analysis on them. But then later, another group take that DNA sequence and put it in a phylogenetic tree with other species, and saw that it seems like it's closely related to human DNA, which is of course impossible, right? Dinosaurs like sister species to human. What this result tells us is actually this was what they sequenced was basically a contamination from the researcher himself or herself. So this is where the ancient DNA studies kind of stopped or slowed down, because everybody realized that contamination is a really big issue in ancient DNA studies. Being able to prevent and distinguish, filter contamination from the ancient DNA itself was a big issue. So many laboratories stopped their studies at that time. Major, big laboratories stayed in the area, and they started to develop both laboratory methods and computational methods to distinguish contamination from the ancient DNA. So as you can see, it made a dip and then started to pick up, but it never, of course, reached at this point where people sequence DNA from millions years old organisms. Okay. So this was pretty much like the first generation of aDNA studies, where people used small single fragments at a time. And they carried high risk of contamination, and they were providing very little information because people were using this maternally inherited mitochondrial DNA. It gives you information about the maternal lineage, but what about the paternal side of it? And so, during that picking up period, people started to develop second generation ancient DNA methods where the genomes come into play during that time. So we were able to start sequencing whole genomes with the second generation of ancient DNA methods. So during that time, the oldest genome obtained so far was 700,000 years old horse genome from the Yukon territory of Canada. This came from a permafrost, which means like frozen soil. So cold is really good for DNA preservation. So if it's cold, then DNA doesn't degrade much. It degrades, but not as much as in a hot climate. So in a sense, in that sense, actually, Anatolia is not in a very good position because we are in a Mediterranean climate where we have hot weather, especially during time. That's why it's not easy for us to obtain ancient DNA in every single material we study with. But anyway, the oldest genome was 700,000. So that was a great success. But then another publication came out just last year on a 1.2 million year old mammoth, again from Siberia, from a cold climate. So this kind of pushed the limits to a much older time. So, and I'm proud that two of our students and Mehmet Somel was in this publication. So they contributed in the computational analysis part of this publication. But now, I mean, the oldest DNA is like a million-year old DNA. So, but again, I mean, this is only possible if the material is coming from a cold climate, from like northern pole, from Siberia, from permafrost, okay? So with the second generation ancient DNA methods, where we can obtain the genomes, we were able to, or we are able to obtain millions to billions of DNA molecules at the same time. So we can use these sequencing machines and like obtain maybe hundreds of genomes in few days. So, I mean, imagine like sequencing a very small part of mitochondrial DNA versus hundreds of genomes in a few days. So we obtain genome level information with the second generation ancient DNA methods. So many variants like positions showing variation throughout the genome can be obtained. These positions are called single nucleotide polymorphisms. And of course, with the second generation of ancient DNA methods, both laboratory methods and computational methods could help us prevent and detect contamination. So we have like special procedures we use in the laboratory to prevent the contamination or minimize the contamination at least. And then with the computational statistical methods, if there is any contamination, now we are able to detect that and then filter it from or remove it from our data. I think Ezgi will talk about this a little bit, so that's why I'm not going into the details of this computational part. Okay, so in our labs in METÜ and Hacettepe, of course, we're using the second generation ancient DNA methodologies, and we are aiming to obtain genome level data. So as Birgül mentioned when she was introducing us in the introduction part, METÜ Ancient DNA Lab was the first lab established, I mean, as far as we know, established in Turkey in 2013. Professor Dr. İnci Togan was the PI at that time, but now she is retired. So Mehmet Somel took over the lab. And we also established another lab in Hacettepe Anthropology Department. So two labs are now like sister labs. We were studying like as one big group and collaborating. So what we do in our ancient DNA labs, first we get the bones from the archaeological excavation sites, or sometimes they send them to us. And then we obtain bone powder from these specimens. We clean their surfaces as much as we can. Sometimes we use, if it's not a really badly preserved bone, we wipe it with bleach to get rid of the dirt on the surface. So it helps to get rid of the contamination, at least to some degree. So we obtain this bone powder, and then we extract, we treat it with some solutions like chemicals and get the DNA out of it, okay? But of course, that ancient, the DNA we obtain doesn't only have the ancient DNA we are targeting. As I said previously, there is bacteria colonized on it, or there is contamination from the modern sources like the researchers themselves. So as you can see here, it's like a mix of DNA from different sources. It's a soup of DNA having contamination and degraded ancient DNA. So as a ancient DNA source, most of the time we prefer the temporal bone, the petrous part of temporal bone, because studies show that DNA is well preserved in that part of the body. It's a very compact bone. Most of the time we aim to use temporal bones, but if it's not available, then tooth are also a good source of ancient DNA. We prefer tooth as a second source. So we extract DNA from petrous bones or tooth, and then prepare whole genome libraries. So those are basically the molecules, all of the molecules, DNA molecules we have in this soup, and then we send them to sequencing. They get randomly sequenced in those machines I showed you in the previous slide. And then we get the raw data, okay? And then after we get the raw data, that raw data is aligned to a reference. If it's a human we are studying, then it's aligned to a reference human genome. And whatever molecules do not align to reference human genome are eliminated, filtered out. Those are the bacterial DNA or contaminant DNAs, okay? But you may ask, how do you know, the modern human DNA, like the contamination DNA, will also align to human genome? Of course, there are some signatures we look for in our data. The molecules that has those biochemical changes I mentioned earlier in my presentation. If you remember, I said there are C to T changes at the ends of the reads of the DNA. So we look for those signatures in our reads. So modern DNA or modern molecules are not going to have those changes, because that happens throughout the time, okay? When the bones sit in the soil for maybe thousands of years, okay? So after obtaining and filtering the data, so what we do is we look for the amount of endogenous DNA, the DNA we are targeting for in our data. So if it's above certain level, above 1% or so, that's good. That's as good as we can get in Anatolia, especially for earlier samples, like Neolithic samples, which are thousands years old. But if we come to a much closer periods, like Roman period, of course, the amount of DNA increases because they haven't been sitting there for thousands of years, like 10,000s of years. So degradation will be much lower. So we will have much more endogenous DNA in our data. So, I mean, after looking at the amount of endogenous DNA, if there is 1% or more endogenous DNA, we use those samples and then we sequence them more to get more quality data. Another way of increasing the data quality is basically, I mean, remember our DNA extraction has, I mean, contaminant DNA, bacterial DNA, it's a soup. So what we do is another way to increase the data quality is going into your DNA and capturing the endogenous or target DNA out of that soup. So we use like special probes to do that. So those are like the probes complementary to the human DNA. So they hybridize and then we can pull them out of our DNA solution. Whatever left is mostly microbial contamination. Then we sequence that captured molecules again. So that way we will be, instead of like sequencing microbial contamination, ancient DNA, modern contamination, we will only be sequencing the captured target DNA. So that way it will be increasing the quality of our data. So that's another way of getting data in our studies. So, I mean, this is something we have done and we are doing in our lab. For example, in one of our studies in 2016, we captured human ancient DNA from our DNA extractions. And as you can see, so this is like the amounts in no capture data. After capture, we have seen, I mean, some improvement. So like up to 4-fold increase in the endogenous DNA content. So that way, I mean, what this does is, of course, I mean, sequencing is a very like very expensive method. So instead of sequencing irrelevant stuff like bacterial genomes or contaminants, we are, by capturing the target DNA, we are only sequencing what we need. So that way, I mean, we are lowering our costs, okay? That's how it helps. It both lowers the cost and then increases the quality of the amount of the data in our studies. So these were the, I mean, like the ancient signatures, I mean, like the biochemical changes and signatures telling us that our molecules are authentic, they're ancient. So we are expecting these changes, C to T changes at the ends around like 20–30% at the broken ends of the molecules. So these as biochemical changes are expected to increase with time. I mean, like in this 2012 study, they compared the amount of like the accumulation of those biochemical changes from 20-year-old sample to 50,000-year-old sample. As you can see, there is a steady increase in the amount of C to T changes at the end. So the older the sample, the more we have these changes in our ancient DNA. So in a like no authentic DNA, we don't see those changes at the end, but in an authentic ancient DNA, I mean, we expect to see these changes in, as I said, 20 to 40%. Okay, so this is how we check the authenticity of the ancient DNA, looking at those changes. Another way is, I mean, if we see, looking at the mitochondrial DNA, we're expected to see only one like mitochondrial DNA type, which we call mitochondrial DNA haploidy. If we're seeing more than one mitochondrial DNA type, then that means there is some external source of contamination in the data. So this tells us that we have to be careful with our data. We have to filter those contamination from our data. So this is pretty much what we do in our studies. So I mean, at least the laboratory part, this is how it works. I think Ezgi will pick it up from here and tell you, like, I mean, I talked about this part, little bit of the computational part, but Ezgi will pick it up and then tell you more about the computational part, and then what we do with the data, like some special cases of our studies.
Thank you so much. Okay. Thank you so much for invitation and this great workshop, actually. At first, I should mention this. So as Füsun said, I will start with where Füsun dropped, so with computational analysis in ancient DNA. Yeah, starting with the same slide. Füsun talked about how they process samples, bones in a molecular biology laboratory, which is, I'm totally a stranger because I'm not a kind of wet lab person. I'm just working on computational part. Yeah, starting with computational part, actually, as Füsun said, after having the DNA out of the sequencing machine, we have some bases, like let's say letters, A, T, G, C, as you see here, and then we align those to genome. How we do this is a kind of computational algorithm, I can say. Let's see, raw data looks like this, which is not important at all, but just I want to make you just imagine what is going on here. That's why I'm showing those samples of files. This raw data comes from the sequencing machine, and then when we have the data, we have just some fragments of DNAs like this. As Füsun said, it's a kind of soup, and we are trying to choose the relevant organism from this soup. Currently, our relevant organism, I will tell all the examples from human, but it doesn't have to be human. We will talk about it later. How we align those, we have actually, after the first released human genome, scientists created a kind of reference genome, and it's updated time to time. It's still, they are updating the reference genome. We call those things as reads, fragments of DNA, they are called as reads, and we just have some algorithms to align those to a reference genome. Just think that we order those reads according to our reference genome, because there are unique patterns on those human genome. Like, let's say there are different orders of those bases, adenine, guanine, cytosine, thymine. We have a certain pattern of those bases on our genome, and according to this certain pattern, we can just match those reads with this reference genome and order those reads. And some of the reads, as Füsun said, they are not relevant to our studies, like pathogens or something else. That's why we filter them out as the first part. What we do actually at the beginning, we just check if we have really relevant organism in our DNA soup, like human. And if we check also if it's like authentic, like it's really ancient or just contamination. We just check this at first. That's why we sequence this first part. We call it as pre-screening. We just sequence a little amount of reads randomly, totally randomly, and check the human proportion in those reads. Then if we have enough proportion, we again sequence it. This time it's deeply sequencing, and we create more data, and we merge all the data using the same procedure like alignment and so on. Aligned files look like this. As you see, I just tried to simulate it here, but the real type is this. And in second generation sequencing machines, we read one base more than once, because there could be errors, or according to our data, like if it's very old, there could be some mistakes in our data. So we read each base more than one, and we create a kind of consensus in the end. Let's see how this consensus actually, according to this reference genome, let's take this part only. That, let's say there is a base here, and we read this base, as you see, like many times. And in our genome, actually, there are different markers. We call them like markers, because we use just part of genome, not whole genome, because we have 3 billion bases in our genome, in human genome. We don't use all bases, because most of the part of the DNA is actually the same for all humans, or even for primates, or if we are talking about, for example, some highly conserved regions like some developmental genes and so on, they are same for all organisms. So we choose the variable sites. If we are working with human, for example, we choose the variable sites in humans, so we can catch some patterns there. So let's say variable sites are those like colored stuff. We pick those parts for one individual here, but we cannot compare one individual by itself. So we create many genomes from different individuals. You will see later. Let's say we have different genomes from different people. And if we have having this, let's say, having this data, we calculate the human proportion. Human proportion is basically reads mapped to human genome divided by all reads in the soup. If it passes certain criteria, for example, if we are working with neolithics, we say, okay, 1% is enough, because in Anatolia, 1% for Neolithic samples is really good. But for Roman, for example, samples, we expect more human proportion. So we determine the threshold according to our sample, actually. And check also authenticity. What Füsun showed, actually, those C to T transitions, for example, or fragment lengths, because during the when the sample is under soil, it's the DNA is not intact anymore, because some bacteria, there are some bacteria like eating DNA, so or just for chemical reasons, DNA is fragmented. So we calculate the length of average length of those fragments, and those lengths also give some insights to us. If we pass this certain threshold for authenticity and human proportion, we continue the rest of the analysis. If we don't pass, for example, let's say we have a Neolithic sample from Anatolia, and we have 1 in 1,000 human proportion, or it's just full of modern DNA, modern contamination, not ancient. So we stop there and get rid of those samples, or we go back to the bone and extract DNA from the scratch. Otherwise, we continue the analysis, like go back to laboratory, molecular biology laboratory, and take the samples for deep sequencing, or maybe if it's a really important sample, we can also capture the human genomes, what Füsun showed. So we send it again to deep sequencing. After deep sequencing, actually we repeat the procedure. We align all the reads to reference genome and so on. And once we align those to reference genome, we just get part of genome, as I said. But what is this part of genome? In ancient DNA studies, the general rule is using single nucleotide polymorphisms. What is this? Actually, those are a kind of substitutions of nucleotides. In human population, roughly, we can say 1 in 1,000 bases in the genome, they are like variable. So they are different from person to person. And from the same population, we can get similar patterns of those orders, like we call haplotypes. But for a distinct group, let's say for Anatolian Neolithic and let's say some Native Americans, there are overlapping regions are not the same of, those SNPs. So that's why we take those SNPs, we call like single nucleotide polymorphism. We take those SNPs and create another type of data, like including only those variants, those SNPs. It's a kind of, let's say, composite file, like including only this information. And what else then, after having those variants, we call it variant calling. After calling these variants from the genome, we actually continue with downstream analysis. What are those downstream analysis? I will talk about them on samples, examples of our work, because just telling some math formula here wouldn't be so meaningful. That's why I will tell those downstream analysis using cases. But what about the actually those filtered out DNA? Are they just trash or something else? No, actually they are not trash. Here we get rid of those data, but we keep them for the later analysis. Like, for example, in the human history, you know, pandemic is not specific to today. In the past, there were lots of pandemics. So recently, actually, there is a kind of developing tradition of working with ancient microbes. For example, what are those? Like plaque. It's the pathogen of plaque is Yersinia pestis, and it was really huge pandemic. You know, lots of people died in the history. So people, for example, extract some plaques from those non-aligned DNA, or some oral microbes, or some other microbes, like which is relevant to history or human or something, you know. So actually nothing is trash. I can say there are some there are some insights into history using those filtered data as well. So let's talk about a bit Anatolia and ancient DNA studies in Anatolia. I will give you examples of our studies, but of course there are other groups working Anatolian Neolithic or later periods. So there is a kind of growing data set of ancient DNA in Anatolia. Here you see the published genomes from Anatolia from different periods. And yes, slide is Turkish, but maybe I can just translate it. We have only unfortunately currently, only one epipaleolithic sample from Anatolia, which is Pınarbaşı sample here. And we have a bunch of Neolithic samples like from Central Anatolia and also Western Anatolia, like Barçın, Çatalhöyük, and Aşıklı, Boncuklu, those are our Neolithic samples published already. And there are some Bronze Age samples and also Chalcolithic samples, which also relevant to Anatolian history. But I can simply say after Bronze Age, or even for Bronze Age, we don't have lots of samples currently. People continue generating the data, but currently published data is mostly focused on Neolithics and partially Chalcolithic and Bronze Age. So, and we have some Ottoman samples, I think currently 3 samples in the data sets. So, what we get actually until the first published sample, for first ancient DNA sample from Anatolia was in 2014, as far as I remember. Since then, actually, there was a growing interest to Anatolian Neolithic and also Fertile Crescent, of course, not only Anatolian. So this gave us some insights into Anatolian Neolithic and later periods. I will just try to give a kind of abstract of those studies. First, let's look at to Epipaleolithic and Neolithic era. This analysis is called principal component analysis. It's a kind of dimension reduction analysis, and it's widely used in ancient DNA research because clustering people according to the patterns in the genome is it's very easy analysis and also gives really good insights. So we generally in our research also practically we start with PCA. I mean, principal component analysis. It's not, by the way, specific to ancient DNA. It's a kind of method used for a really long time. For Epipaleolithic and Neolithic area, if we look at this picture, this figure, those gray pale dots, let's say, they are the modern genomes. What we do in this kind of analysis, since we have a data which has some missings and some it's not like modern, you know, since it has lots of biochemical changes and stuff. It's not like modern genomes. Modern genomes we can read the modern genome many times, and also we can get really good data. But for ancient DNA data, we have lots of missings according to the data quality. So we need to handle those issues. What we do in this kind of analysis, we create a kind of baseline using modern genomes without missing data or some trash in the data. We create a layer, baseline, and then we project ancient samples onto this baseline. So we create a kind of diversity of modern genomes, and we check how our ancient DNA data behaves in this diversity. As you see here, this is a kind of Western Eurasian population PCA. Those are just Western Eurasians. I can just navigate you like those are Levantine modern samples and going to the Caucasus here. And here there are Southern European samples and Northern European samples. This PCA shows a kind of geographical distribution. And when we project our samples onto this PCA, as you see here, Anatolia falls, Epipaleolithic and Neolithic era of Anatolia falls into here. But this is not, we cannot evaluate our data by itself. We need to compare it with other samples from the region. For example, we know from archaeological part, actually we have lots of connections with Levant and Zagros, Caucasus in the history. So we check how it changed during the time. As you see here, actually here there is Epipaleolithic Anatolia, and here there is Epipaleolithic Levant and Epipaleolithic Caucasus. And there is a kind of triangle during the Neolithic and Epipaleolithic, and others fall into this triangle, which means actually we can say there was human mobility during the time. So this mobility make people maybe closer or something. We are checking this. As you see here, from Epipaleolithic to Neolithic, Anatolian samples comes through the Levant. It's closer to Levant. You can just think that those points, closer points, are closer genetically or vice versa. Basically, in Epipaleolithic and Neolithic, we have a kind of triangle, which Levant and Anatolia has some connections, but also there are some connections with Zagros and Caucasus, which moves also a bit through the Zagros. When we come to Chalcolithic era, Chalcolithic samples falls in between, those Anatolian samples fall in between this triangle. We can say, and different, actually geographically, there are different affinities. As you see here, for example, those are Central Anatolian Neolithic, those green ones, and those are Northern Anatolia, I think İkistepe, Chalcolithic, and those moves to Caucasus a bit more than Central Anatolians, for example, and so on, actually. There is a kind of homogenization after Neolithic, we see. Maybe this is a kind of indication of mobility, post-Neolithic mobility. And in Bronze Age, also we have similar pattern with Chalcolithic, because this mobility continued at that time. And in Iron Age and Ottoman era, it's like, there are different patterns of mobility, as you know. For example, for Ottomans, there are some Central Asians coming into Anatolia. So the pattern of PCA also changed. For example, those Ottomans move to a bit different way. Actually, this we know from the literature that this is the pattern of Central Asians in Anatolia. And there's one European guy here. So, what are our studies in Neogene? We use ancient DNA data in different to solve different solutions, not like only population mobility or something. Like we also make kinship analysis and some other analysis, which I will show. So we get insights not only into population history, but also like some social issues, like kinship, social kinship, or biological kinship, which was a debate in anthropology for a long time. So one study that we published last year, which was focused on kinship, and we published for the first time Aşıklı genomes and Çatalhöyük genomes. And in this paper, actually, what we did, we just check the patterns of kinship on site, let's say, like because as you may know, many of you may know, those people in Neolithics in Anatolia were buried under the floor of those houses. So we were curious if those people buried in the same house, which we call co-burials, if those co-burials were biologically related to each other or not. And when we check this, actually those Aşıklı and Boncuklu samples are from pre-Pottery Neolithic, and Çatalhöyük and Barçın are Pottery Neolithic, so later periods. What we see in Aşıklı and Boncuklu, the kinship, biological kinship, were like more co-buried individuals, among co-buried individuals, but for Çatalhöyük and Barçınhöyük, they were not like that. Close kins in the data were not buried in the same in the same house, under the same floor, for most of the case, let's say. So what we see in the frequency of covariate clusters, there were, like, we compared it statistically. So in Aşıklı and Boncuklu, I mean preparatory Neolithic, most of the kins were covariate individuals, and in Çatalhöyük and Barçınhöyük in the later period, they were not covariate. So we don't have any reason to think that the kinship patterns in Neolithics should be same as now, as like core families or something. There's no reason to think like that. So we had a kind of insight to this, like in pottery Neolithic, there was a different kinds of social relations in those sites, at least in Anatolia, both in central Anatolia, represented by Çatalhöyük, and also Barçın. And another paper last year came out from our group, was checking the runs of homozygosity. What is this? Actually, in the genome, if you have, like, let's say, we would expect some variability in a genome, but sometimes both alleles, which is like one comes from mother side and the other one comes from paternal side, they overlaps each other and they don't have variability. They have the same bunch of information. We call this runs of homozygosity, and if those homozygous sites on the genome is really long or distributed and they have lots of homozygous sites, we can measure the consanguinity in those genomes. As you see here, for example, those are, there is a kind of example of cousin marriage here, those DNA comes from grandmother and grandfather, and the haplotypes of those individuals are like you see as colored, and those are, there is no consanguinity here, but there is consanguinity here, as you see. And this guy would have, since these and these are cousins, they would have some sites overlapping, as you see, same color, overlapping sites with the same haplotypes. So when we check those haplotypes, the length of those haplotypes or distribution of those haplotypes, we can guess the homozygosity, or it shouldn't be, how to say it, it shouldn't be like not only consanguinity, but if we have a small population, all the reproducing child would be like those partners would be a kind of kin, maybe closer kin or further kins, but they would be a kind of kin. So it would also create some rows in the genome. So we guess which one is the case, like consanguous matings or small population size. So we published a paper again last year, which these rows were in charge. We compared the population, let's say individuals having long rows or something. So what we simply say from this data, from hunter-gatherers, starting with 12,000 BC, we see a decrease of runs of homozygosity in the genome, which means basically is a kind of increase of population size in the throughout the history. But there are some outliers here. Who are those? We checked those. Actually, this is the published Ireland individual from like a kind of tomb in Ireland, Neolithic individual, middle Neolithic individual, which they publish it as like, how to say it, first degree kins mated, and this is the child of this first degree mating. We checked this also with our method, and we saw, yes, actually, those are simulations. We simulated some data, and according to those simulations, really, this is a kind of incest child. So there were some cases in the history, like extreme close kin matings, but mostly it was like more like depending on the population size. And as I said, we can see the traces of increasing population size through the time. Actually, there are traces in the genome, human genome. Only human, like as I said at the beginning, we are not dealing with only human. We also have other species, like again last year we published Neolithic sheep samples from Anatolia, because it's not only about human, but also it has some connection to the culture, because Neolithic sheep are actually the topic of cultural evolution of human, because of the domestication, because we know that sheep was domesticated in around somewhere here. So we were like searching the origin of domestic sheep, and basically we realized it has really complex history, not like a kind of simple history, but it has complex history. I just wanted to touch this. I will not give the details, but we can discuss this Q&A session. So recently we published a new baby. It's like currently it's preprint, not published in a journal, but I just wanted to give a small insight about it. Çay is very important for us, because there is no published genomes from Upper Mesopotamia. I mean Neolithic samples. Upper Mesopotamian Neolithic samples are a bit unlucky, as you may guess, because this part of the world is very hot, and preservation of DNA was not good. But thanks to our great wet lab team, we managed to get some data from Çay, and we continued producing new data, and we got some insights how those people look like during the Neolithic. We got some insights. I can give just a small brief explanation about it. As you may get used to this PCA, it's the same gray dots as previous, and those are Neolithic samples, again like Anatolian Aegean samples and Levant samples, Epipaleolithic and Neolithic, and again Zagros and Caucasus. And Çay individuals, like those red stars, fall in between those groups. Before Çay, we didn't see any group falling in this line, like that much Zagros-like individuals. So it was really interesting for us, but also it's not quite strange, because Çay is the middle of, geographically is the middle of those triangles. So it reflects the geographical position in the genetic data. This is another kind of dimension reduction analysis, shows the similar thing. We have, as you see here and here, we have one outlier. This outlier individual, actually, what we investigated is like, it's a recent ancestor of this outlier individual came from Zagros, which shows the mobility during the Neolithic also. yeah, this is a kind of multi-dimensional scaling analysis, which I will not go into detail. What else we do with this data, not only PCA and MDS, of course, or kinship analysis, but also we use this data to estimate the proportion of different source populations, like all our genomes are admixed. I mean, they are composite genomes, and we can investigate which part of, not which part, but how much proportion of our DNA come from one source and how much the rest. So this is a kind of admixture analysis we can call. And using those methods, there are several methods. I'm showing here QPADM method, but there are several methods, and we can estimate the proportion. For example, for Çeönü, we estimated that a big portion of genome comes from Anatolia. And there are some Levant and Zagros Neolithic traces in this genome. And for the outlier individual, this is those Çeönü samples population. This is outlier individual, and this outlier individual can be modeled as Çeönü population plus some Zagros contribution. And one of the striking results of this study was actually how Mesopotamia influenced the later populations, like later PN, Proto-Neolithic populations or Bronze Age populations, because there was a kind of hypothesis that maybe later populations, homogenization in later populations came from like Mesopotamian influence. So we tested this, and what we find out here that actually we checked this, I will not go details of analysis because of the lack of time, but we checked if Çeönü is closer to one group like Anatolia or later Anatolian populations. And here, as you see, we check if this influence comes from directly Mesopotamia or directly Caucasus. So what we see actually here, Çatalhöyük, Barçın, and Tepecik Çiftlik, those are Neolithic, Proto-Neolithic individuals in Anatolia, and those are closer to Çeönü population, like later population is, just a minute, yeah. Later population, the color coding shows the time of the dates of those populations, and later populations are more like closer to Caucasian sources than Mesopotamian sources. So obviously there was something in Proto-Neolithic influenced by Mesopotamia, which we see actually in material culture as well, which Çeönü, some similar aspects of Çeönü and Çatalhöyük or Barçın. But for later populations, like Chalcolithic, starting with Chalcolithic, this is really early Chalcolithic, starting with Chalcolithic, later populations were not influenced by Mesopotamia, or alternatively, maybe after the Proto-Neolithic, Mesopotamia had a kind of population change, and this influenced the later populations, but not preparatory Neolithic Mesopotamia. This is another similar analysis. So I was told that adding a kind of abstract in Turkish abstract at the end of the slide, that's why I added. Should I give a small abstract in Turkish, or is it enough just adding this?
Oh, thank you. Maybe you can say it in a few sentences in Turkish as well. That would be great.
Okay. Evet, Türkçe'ye hemen dönmek çok zor oluyormuş. Şöyle, aslında bizim bu yaptığımız çalışmalarda biz sadece popülasyon yapısını incelemiyoruz. Yani popülasyon yapısı, demografi, benim kendi kişisel ilgi alanım daha çok popülasyon yapısı.
If it is okay, I will use the full presentation mode because I want to go in between the slides through the presentation.
That's absolutely okay if it's for you.
Okay, so I named this presentation Questioning the Neolitization of the Aegean because there's this ongoing discussion about whether the Neolitization of the Aegean, especially the Western Anatolia and Greece, is a result of a major demic diffusion from the core areas of the Neolithic or if they are an indigenous development. So this is one of the major questions that we are actually trying to answer through the ancient DNA studies that we conduct collaboratively between METU and Hacettepe teams through these funding that we received from the European Union projects, twinning projects, and ERC, European Research Council projects. And as Ezgi mentioned at the end of her presentation, we can do a variety of different, test variety of different hypotheses for a variety of different time periods. But for the Neolithic in specific, we have been trying to understand the social structure, changes in social structure during this major transformation in human history from hunting, gathering, or foraging in general to agricultural communities. So this took thousands of years. Obviously, it's not a one-night event or a short time event. And during this time period, belief systems, social structures have changed in relation to the changing needs, economic needs of the societies. So it's then very important to understand this change in general because there's a major change in social structure, and we have to look at how we define the Neolithic process in Southwest Asia in general. For this purpose, I have written here a very general description. It's very difficult to put it in one sentence, but and also the history of research in the area shows that the definitions of the Neolithic changes through time from the early 20th century to this first quarter of the 21st century. We have looked at the Neolithic process from a variety of different angles, and today we understand that it's actually a gradual adoption of long-term sedentism first, and then together with this, a subsistence economy based on, increasingly based on domesticates, plant and animal domesticates, and a social life centered on so-called house in context of the climatic conditions of the final Pleistocene and the early Holocene, which is about 12 to 14,000 years ago. So the early evidence for sedentary life in this region goes back to a better climatic conditions of the final Pleistocene, where we see the first instances of hunter-gatherer sedentism in East Mediterranean, in the Natufian culture. And then this develops, I'm not going to go much into the detail of this, but it develops then into these increasingly sedentary communities. And the primary zone of this neolithization encompasses the Levant, the Taurus-Zagros ranges of Mesopotamia, Central Anatolia, and Cyprus, actually, from almost to get the beginning of this process in this area. So specifically in the aftermath of the Younger Dryas, which is a cold period after the Bolling and Allerod, basically a short going back to the difficult conditions of the Ice Age, sedentary communities began to be established across this zone toward the end of this, in this zone, and during the PPNA, and the first indications of plant cultivation also began to appear. But this doesn't mean that these were domesticated yet or domesticated yet. So between, after this time period, you know, toward the end of this, but we call PPMB, pre-pottery Neolithic B period, and don't worry about these cultural terms. The community sizes began to be increased in these regions, in these settlements, and the subsistence economy based on domesticates gradually became the norm toward the end of this time period. And this is the end of this time period is perhaps best represented in the very much investigated site, Çatalhöyük, site of Çatalhöyük by 7000 BC. We see there a fully established farming village economy with some of the first pottery and related material culture. So the zone that we talked about, you know, obviously this is a zone where these early proto-domesticates, let's call them, wheat and barley and cattle, pigs, sheep and goats, sort of conglomerate of the proto-domesticates. So it's not a surprising thing that we see these first settlements in these regions of plenty, environmentally allowing regions. So this is sort of the PPNA time period, and this is also the time period where we see this Göbekli sites such as Göbekli Tepe, monumental architecture, and burial practices, you know, focusing upon the skull cult, you know, the skull removal and such, an variety of architectural practices in a variety of regions. And in the second time period called the PPMB, as I said, sort of the social structure is becoming more and more visible, this house-based social structure. We didn't know in the past whether these houses actually represented kin-based families or not, but we were, you know, presuming this, and it's becoming more and more clear with the DNA studies that these hunter-gatherer communities, the house structures of the hunter-gatherer communities especially, appear to be more kin-based. However, the later structures, you know, later period, Çatalhöyük houses may have included other individuals from closer groups still, but not necessarily first-degree kin-based. So this is the area where we see these first domestication and sedentism practices. And however, in the Aegean, we see the similar practices appearing only starting after 7000 BC, especially, you know, emerging around 6800 BC to 6700 BC or so, and earliest ones are known to be so far at Uğurluhöyük in Gökçeada, where I also excavate, and also in Ulucak and a few other places. So looking at the situation in this area slightly earlier, during the PPNA and PPMB of East Anatolia and the Levant versus Aegean, it's not that we don't have any places over there. There are hunter-gatherers existing in the Aegean, obviously. However, and we do know these places from a variety of places, from, for example, Bozburun, where I also survey, in Karaburun, where other Turkish colleagues continue to survey, in Kerame, Marulas, Frankti, especially Sarakinos, Cyclops Cave, and a variety of places, as well as in Girmeler and Belbaşı and in Antalya. We do know that these sites in the Aegean and Eastern Mediterranean have both the Mesolithic and the Neolithic layers. However, none of them in the Aegean have so far been successfully demonstrated to show a continuity from this early Holocene to the middle Holocene, to the so-called the Neolithic period. So this has always created a major discussion, whether the Neolithization of the Aegean was a result of actually endemic diffusion from the East or an indigenous development. So this has remained a major issue. You know, outside of the social structure changes in social structure throughout the Neolithic process, this is one of the things that we have been exploring. And archaeologists in the past have used a variety of different types of methods to understand what was happening, and the concept of this so-called, let me show this first, the Neolithic package has been very influential in this. And basically what archaeologists have been doing was that we were following a variety of material culture and also zoological and botanical remains through time and place, and how they are distributed from that core area of Neolithization, and with the assumption that if we trace their emergence, their first place of emergence in the East and gradual distribution through time toward the West, then we will know how the communities migrated. I mean, there was this assumption that if we follow this, it's migration, definitely, because, you know, this is what we were thinking. And so the major thing is follow the obsidian distribution or specific types of lithics, distribution of specific type of lithics, pottery, figurines, architectural practices, domesticates, and such. So, for example, pottery has become an important thing among those. You know, it's it's first appeared in the East. You know, I've shown Çatalhöyük as one of the first places, but this is the core area of the first emergence of early pottery, about 7,000 to 6,600 BC, and then we see it also approximately starting from 6,600 BC or so in the West as well. In very similar forms and shapes. Similarly, figurines, or figurative objects that we call figurines, also seem to appear first in this region in the early Holocene, and then they are distributed, similar types are distributed all over the place, again, together with pottery, especially around 6,600 BC or so. Obsidian distribution has been another thing, because there are different sources of obsidian. So people have been looking at these, and they were saying, you know, the Aegean and the Anatolian obsidian spheres are different, and they are utilizing their own obsidian sources. Therefore, we should look at these as separate cultural spheres. However, again, in this Aegean area, there's this, you know, combined place that we are, you know, that can include both the Aegean obsidian from, for example, Melos Island, as well as the Central Anatolian, especially Cappadocian sources. It's never been easy to understand looking at the obsidian from the perspective of the lithics. Sickle blades, especially these agricultural community, you know, blades that are used for by agricultural communities, are also traced, and their first instance of appearance occur during the early Holocene of the East, but they appear much later in the West. So people have assumed, together with pottery, figurines, obsidian distribution, and these distribution of sickle blades, thought that, you know, there must be some kind of endemic diffusion from the East. And for example, these people, you know, there are, you know, Mehmet Özdoğan, Barbara Horej, Lüning, Gerbold, you know, variety of people have been modeling this neolithization, Aegean neolithization from the perspective of endemic diffusion from the core areas, merely based on the material culture evidence. Yet, I have to say, archaeologists can never be sure about the nature of interaction that caused material culture similarity in time and space, unfortunately. Is the similarity a result of knowledge or an idea change, or is it an outcome of material exchange, or is it an outcome of some kind of population exchange, such as migration, or maybe a smaller scale exchange, such as partner exchanges, or some combination of the above? It's very difficult to understand without, for example, DNA analysis. Testing these hypotheses via archaeological evidence is not easy, I have to say, always interpretive and dependent on your methodological approach and assumptions. So despite this fact, the endemic diffusion hypothesis has been the most popular and easy interpretation for the neolithization of the Aegean and Europe in general. And we do know that there are reasons, for why this has been so popular. There are arguments in favor of this hypothesis, as I've shown. The first Neolithic settlements in the Aegean date back to a later period than that of the East. And also there's no continuity from the so-called Mesolithic or the early Holocene to the later Neolithic of these sites in the Aegean. So these are the arguments in favor have been, and they have been put in a pedestal, so, if you will. But there are problems with endemic. There has always been problems with endemic diffusion hypotheses. For example, the earliest levels of the Neolithic settlements in the Aegean, they do date back to the 6,800 BC or so. However, for example, Uğurlu and Ulucak, however, at these sites, pottery and other important items of the Neolithic package do not occur for approximately 200 years until 6,600 BC. So the question comes to mind, if they are migrating, are they not carrying their cultural knowledge of pottery making, figurine making in this first instance of settlement? But what is happening during those 200 years, and why do we not see that elements of the Neolithic package for at least 200 years? So this is a major question that's still waiting to be answered, and hopefully we are going to be answering some parts of it through the DNA analysis from sites, comparing information from sites such as Girmeler, an early Holocene site in the Aegean, or Ulucak or Uğurlu, actually. And the other thing is some Aegean early Holocene and Mesolithic groups, as information comes through over the last 10 to 15 years, we do know that they, during the early Holocene, those forger groups in the Aegean in the early Holocene display some similar architectural and burial traditions that are very similar to that of the Anatolian and Levantine PPM counterparts at about the same time period. And we also have found some of the earliest evidence from the Bosborough prehistoric surveys that the Melos obsidian, it's an Aegean sea obsidian that was present, may have been present on the Anatolian coast as early as the very early Holocene before the Neolithic. So that changes the game. And if we do have some kind of, if these information, the existence of burial architectural practices at places such as Marulas and Girmeler, that are very similar to that of, I don't know, the Central Anatolian sites of Boncuklu, Aşıklı, or other East Anatolian PPNA, PPMB sites, then we are looking at some kind of interaction, possibly through the sea routes or maybe even through the land routes This interaction is changing the game. Then we have to start thinking about, again, whether we have the demic diffusion or not, really. And we have to problematize these issues that are sort of being hidden from the discussion, actually. So this is, again, this is the slide that I've shown earlier. Ultimately, the recent archaeological research indicates some sort of interaction between the Aegean, Central Anatolia, and the Levant, at least by the late Pleistocene, early Holocene, both through sea and inland routes. So we do ask this question again, what do we have? Is this demic diffusion or not? And I have to say, Ezgi has shown the general information. I have very brief things that I want to show you from the published sites that I specifically indicate. This is the Anatolian gene pool from Pınarbaşı to, let's call it Tepecik Çiftlik, the latest of the Neolithic sites. And within this, we have to realize, Anatolian gene pool is indigenous. There's an indigenous development during the from the Epipaleolithic to the end of the Neolithic. So it's not like a migration from anywhere else, but an indigenous development of the Neolithic in Central Anatolia can be observed. However, for the Aegean, what we have is in some places, such as, so far the evidence shows that for example, in Serbia, Lepenski and Ravenna and Alapitra, or places in Balkans and Italy, Bulgaria, Croatia, Macedonia, Hungary, Serbia. The first two instances of the Neolithic sites show that their gene pool is also very similar to the Anatolian gene pool, or let's call it the Aegean gene pool. While there are other places that indicates the mixture of these events occurring in some of the early agriculturalists of Bulgaria, for example. What we have is Natufians and Western hunter-gatherers, which are the European hunter-gatherers, and Iranian pre-Patriarchal individuals were consistently more similar to the Ravenna individual than those in Barçın in one of the articles that we explored this. So when we look at the situation, we have, I am not going to summarize the article, but this article actually by Kılınç et al. Questions whether there is a common Anatolian Aegean gene pool prior to 9th millennium BC. And we are looking at certain sites and comparing them and showing that there is already, there must have already an Aegean and Anatolian common gene pool occurring sometime during the post, so-called final Pleistocene or Epipaleolithic at the latest, which may be which should be reconsidered in the light of this evidence, because then it's not going to be very easy to talk about actually whether about migrations from Anatolia, because there's a common gene pool here. So they may have gone from anywhere from this area to Europe or other places. So this mixture, these questions are on the ground, and Füsun Hoca, Ezgi, Mehmet, and Yılmaz Hoca and Elif Hoca are exploring these questions. And we do have lack of Mesolithic and early Neolithic DNA from the Aegean so far. Girmeler cave is a very important site, and currently our team is investigating the DNA, some DNA from the site, and hopefully it is going to give some important information about the Neolithization of this region. So that was the case study that I wanted to talk about. I am very sorry that we passed our time period here, but thank you very much for your patience and interest in this lecture.